So, welcome to our final presentation about the Bee's Knees project. And we'll be going over the overview of the project and some of the experiments that we went over throughout the past 2 weeks.
We seem to be having a few technical difficulties.
We hope that you will be patient with us throughout this time as we solve this issue. But we'll be right with you in a very short moment.
Um, Also, Elijah, my screen share was fixed, so
Oh, okay, got it.
take over.
All right, sounds good. I'm sorry.
Hello, can anyone hear me now?
Yes, we can.
Oh,
The screen did.
I'm so sorry. Okay. Let's start over. I'm so sorry.
All good, all good.
All right, is everyone able to see the screen?
Yep.
Yes. Yeah.
Great.
It's not in present mode.
Oh, and we can go, we'll start right off from the very top.
Don't worry, y'all. We'll fix this all in post.
Thank goodness.
Thank you. It's very much appreciated.
Yeah.
Chris, so your computer isn't in present mode. At least it's not showing that screen share. Okay.
Oh, there we go. Sounds good.
So I had
Shake it off, and YouTube video starts now.
Right, so hello everyone. This is our presentation on our modeling project that we have dubbed the Bee's Knees, and I'll start off with the context of our project. Right, so there's a parasitic relationship between honeybees and the Varroa destructor, which is a mite, and the Varroa destructor latches onto a bee's body while the bee is scavenging out for pollen and interacting with flowers, entering a bee's hive where it can reproduce. And if we look at this simple drawing diagram here, you can see the macro scale perspective of this system, meaning how the mites interact with bees outside the hives and how multiple hives can transfer mites between one another. So if you look at the start BA here, you can see that it can take the white path of arrows, go to a flower that has a mite on it, and it can either take the pink arrow path and return to its hive where the mite can go and enter the hive and reproduce, or it could go to another flower following the blue arrows and drop off a mite and return to its hive mite-free. This meaning a bee from a second hive, start B here, could follow the white path and grab that mite and take it back to its hive, and this cycle can continue between multiple hives. If we go to the next slide, you can see the microscale interaction between mites and bees, where within the hive, the varroa mite can ride on a worker bee to reach the brood or larvae within the cells. They enter these cells and feed off the bee larvae, producing approximately 2 mites per brood. While they're feeding on that larvae, they transmit diseases into it. And this disease that we will be focusing on for our project is the deformed wing virus, which wrinkles up the bee's wing and limits its ability to fly. Once the larva fully develops, the capped cell becomes uncapped and the larva leaves the cell. And with that larva, the mites also leave the cell and will attach to a worker bee that brings nectar to a new larvae. And then that cycle will continue where they go and enter the capped cell and reproduce more. And that results in exponential growth of the mite population. So we decided we wanted to look into essential oils as a treatment to prevent this growth of population of mites within hives. And one of the essential oils is thyme, which affects the macro and micro scale relationship by disorienting the mites and blocking their pores, causing them to fall off bee bodies. This results in a decrease in the number of mites within the hive and a decrease in the probability of the mite attaching to bees at flowers. We also wanted to look into spearmint, which affects the microscale relationship by decreasing the transmission rate of diseases into the larvae, and this results in a decrease of bees that have contracted the deformed wing virus. To get a better idea of what this looks like for our macro scale, if you would click on the next slide, we drew a compartment diagram of the states that bees can change, and it can be a susceptible adult, and once a mite gets onto it, it's called an infested adult. And when we implement our time, that adult can return back to its susceptible state. And as for spearmint, a spearmint oiled adult can still be susceptible to having a mite add onto it, but it won't become sick. We also made a compartment diagram for the mites for the microscale, and we just decided that there would be two states for the mites where they can be reproducing in a capped cell inside the hive, or they can be outside of the capped cell within the hive, traveling to a new capped cell. And there's multiple ways that more mites can be produced, or they can die off or be knocked off by time. And that just results in 6 to 8 mites being produced per mite. So now we'll talk about our model.
All right, so this is kind of a complex system to model. So we kind of broke it down a little bit. So what are the key players in this kind of problem, right, between the varroa mites and the honeybees? So the first, of course, are the honeybees. Specifically for this model, we use the Western honeybee, modeled our specs off of that. The second are the hives, which the bees return to, and where they breed and raise offspring. And then the third are the flowers, which are the bees' food source. So, all in all, this is a simple screenshot of what our ecosystem looks like. So here you can see there are 3 hives and lots of honeybees. Each honeybee is assigned to a specific hive as its home hive. All right, so in order to model some of these interactions and kind of model this whole system, we implemented a couple functions. So the first of these is reproduction. So obviously bees reproduce, and within our problem, actually reproduction was especially important to model because the varroa mite disease, so the deformed wing virus, is transmitted to larvae, not adult bees. Adult bees can only carry the mites back to the hive, but only the larva can contract the deformed wing virus. So this was especially important. And so here you can, in this animation, you can see that the population is increasing. So the second function that we had was energy. Now, energy in our model dictates essentially how far a bee can forage. And if a bee runs out of energy, then they die. And then so this we use to model kind of how in bees with deformed wings, they can't forage as far because their wings are either reduced in function or non-functional. So they would have a lower starting energy. Thirdly, flight patterns were another function we implemented. This was just to code the bees to be able to, first of all, know which hive they belong to, travel to different flowers, and based on the amount of energy they had, decide whether they wanted to forage from a flower to another flower or to return to the hive. So we had a couple probability-based functions to illustrate the entering and exiting of mites within our system. So the first way that a mite can exit the system is to fall off naturally, like from a bee, and this was kind of determined randomly based off of random probability. The second is grooming. So when a bee grooms, the mite can be knocked off. And the third is when a bee is flying, the mites can also be knocked off. And these all exit the mites from our system. However, so the main way that mites enter the system, besides reproducing within the hive, are to be contracted from flowers. So when a bee lands on a flower, there's a probability every time that a mite might kind of latch onto it, and then it'll enter the hives. So, let's take a look at kind of the different states of bees that the bees can be in. So the first one is just a regular yellow bee, honeybee. This is a bee that is susceptible to deformed wing virus. It is susceptible to being latched onto by a mite. Here we can see, you can kind of see here there are some red bees, and those indicate that those bees are sick with the deformed wing virus, or they are infested with the varroa mites. So here you can see these blue bees are somewhat protected from from being infested by mites. So so in this function, bees with the time, it decreases the mite population and also decreases the probability of the mites actually being able to latch onto these bees. And then lastly, our spearmint function. So you can see the cyan bees, those decrease the bees that actually contract the deformed wing virus. So we know that there are some assumptions and limitations to our model that are important to bring to you guys. So the first is time scale. So we tried our best to our each tick is 30 minutes. So we tried our best to kind of model the speed at which the bees travel. However, there were a couple of things that were not really realistic for us to model. For example, typically a hive will take 2 to 3 years before any noticeable signs of deformed wing virus or other viruses transmitted by varroa mites is really noticeable within the hive. And however, considering our computational ability, it was just not realistic to have that many to run the model for that long. So another limitation we had was with bee reproduction. So as some of you may know, there are several stages of life for a worker bee, which not only include exiting the hive and collecting nectar, which is the foraging stage, but there are other stages that we didn't model just because it was too complex for our model to handle, and also it was not extremely relevant for the specific problem we were addressing. The bee population size is also not necessarily representative of an actual hive. An actual hive will have hundreds, if not thousands, of honeybees. Obviously, we do not have the computational power to really simulate that, so we reduce the number of bees. And then lastly, we made an assumption that weather was consistent and optimal, so like summertime weather, throughout our simulation. Even though there are kind of variations in some of these parameters and specs during wintertime, but we were not really able to implement that into our model.
All right, so from now, me and my colleague Eli will guide you through some of the experiments we did. So first we use the same setup for all of our experiments for to be consistent. We used 10 repetitions for each iterations for good measure. The total steps or ticks each run did was 1,500 or 3,000. With the tick being 30 minutes, this would translate to 125 days and 250 days respectively, which sort of fits about a season, say summer, so we don't have to account for the weather. The response variable, we decided to measure were the percentage of sick bees, since this has most effect on the hive and the workload of the hive, and also the total amount of mites in a hive. We first vary the number of the starting mites, which are the mites that are on the flowers and then latch onto a bee and then go to the hive. We weren't surprised. This is a pretty linear growth. Like the more mites you have in flowers, the more chance there are they go to the hive. One future direction we could take this to is look into the use of pesticides, which harm mice but not bees. Again, this would just affect the speed at which the population growth. It's not a very optimal solution for completely healing the hive of mites, it would just decrease the speed at which the mites grow.
Moving on from that, we decided to, change up the number of flowers in the environment just to see whether or not the flower availability in the environment would really make a difference in either the percent of bees becoming sick after the period of time or the total mite count after the set period of time. But as you can see here, after varying the number of flowers in the environment from like 0 to like 50 flowers in the environment, there wasn't a really significant difference, or just like as you can see in the data, there is no real correlation there. So the main takeaway we got from this experiment was that there was no correlation between the two. However, if we move on to the next experiment, when we start really thinking about how our treatments come into play here and like what we can test about using the different kinds of essential oils, what we found was that if we were to change the time in between the reapplication of these treatment oils or these, for example, in this case, time, we wanted to see whether or not there would be a correlation there. And as you can see on the left graph, there doesn't seem to be really a correlation there. It seems to be kind of random whether or not it like completely prevents the bees from becoming sick or if it completely makes the entire hive sick. So we won't talk about that here. But as you can see on the right graph, we were able to fit a somewhat of a logarithmic model to it, and like here's a rough curve for you just to like make sure that it's a little clear about like what we're referring to as the logarithmic model. But as you can see, what we found through this experiment was that the more time you put in between reapplication of this time oil, the more the mite count increases over time. So so one of the future like directions we could do based off of this experiment is to further like refine the model and use this model to really find the cutoff time, which would keep infestation rates low, or below, at least below the suggested 3% infestation rate according to source.
In addition to time reapplication, we also varied the spearmint reapplication, and this is just like amazing. So the spearmint does a pretty good job at keeping the sick bee population under 20%, and it's still linear, but it doesn't really vary. Of course, the optimal solution to keep the bees healthy would be to continuously apply spearmint, but that might not be very cost efficient, especially for farmers. But as you can see, keeping it at like 200 ticks, 400 ticks, which would translate to about 16 days or a month, it doesn't really differ much. So you're not getting any too much sick bees from it. And we also decided to do the combination of spearmint and thyme, and this was actually surprising. It does worse than just applying straight thyme. There's a few issues with this. First, because of our lack of time and computing power, we assumed we applied both spearmint and thyme at the same time. But that might not be optimal. Maybe applying thyme more often and spearmint less often would be more optimal. But again, we didn't have that sort of computing power.
Really, to just kind of wrap up like our discussion of our experiments and to talk about to reiterate on some of the main takeaways that we got from the experiments. First of all, we found out that like through the linear trends that we found with the starting mite count, we found that our model somewhat reflects the behavior that we would expect in this kind of biological systems, where like if there are more mites that are introduced to the environment, then it makes sense that the total mite count and the total percent of bees would also increase linearly with that. In addition, we learned that some other variables like flower counts, availability doesn't really affect any of the response variables at all. However, we did find that first we found a logarithmic trend in varying the time reapplication times. But we also found that in the spearmint reapplication, not only did we find a somewhat of a linear trend in both the percent of bees sick and the number of mites after the set time, but we also found that spearmint also somewhat performs really well no matter how long you no matter how long you spread out the time of reapplication of spearmint. So one of the bigger conclusions that we wanted to make from our observations here was that spearmint works as the most effective essential oil treatment in both the disease transmission intervention and hive mite control. And so finally moving on to the implications and the takeaways of, of our model, we do recognize the fact that our model isn't perfect. There are still a lot of things that we could go back into and make and further refine just to make sure that the outcomes that we do get are better. But as for now, at the current state of our model, we know for sure that we can say, based off of our model, that essential oils are effective in combating varroa mite infestations and just somewhat preventing the bee disease transmissions as well. And we also found that spearmint treatments, like if you had to consider between thyme and spearmint treatments would most likely be the most effective in terms of keeping the varroa mite infestation damage as low as possible. But in addition to this, we know that in real life, we know that actual bee farmers use a mixture of these essential oils, and because it just makes sense in general. But in addition to this, although we weren't able to really thoroughly investigate like the mixture of essential oils really in-depthly, we do acknowledge the fact that there is a lot of potential in not only finding experimenting with the different like ratios of essential oils, but also during the different times between reapplication of these essential oils. But in addition to this, there's so many essential oils out there that we could have tried, we could have implemented into the model, but we just didn't have enough time to. But we do believe that with the model that we have right now, with more time and effort and just more fine tuning of this model, should be able to find the most effective mixture of essential oils that will help, that may or may not help bee farmers in the future create a cure-all cocktail maybe in the future. But other than that, that is the main takeaway from our presentation. But we do have one last thing for you guys, because we do know that we have a track record of doing something. So I'll let my colleague Carissa to introduce that.
Sorry. So this is plus Ryan. Okay, so basically, as you all know, we always include a reference from the Bee movie. However, this time we decided we wanted to pay homage to our wonderful, wonderful organizer. Okay, so introducing Michael Barry B. Benson Babine. So you guys know Barry the Bee, and then so we decided to just pay homage to the wonderful Dr. Levine, and plus also Ryan, our mentor. So sorry, Ryan, couldn't find a photo of you, so we couldn't turn you into Barry the Bee. But thank you so much. This was an amazing experience for all of us, and we just decided to implement this photo into our model. So Barry Benson Bavine. Yeah, so thank you so much, and we love this opportunity.
We hope you have a blast.
Any questions?
Wow. At least this time I wasn't being consumed by parasites.
You were
Thank
just
you.
a
Y'all
field
are
of
so,
flowers.
that's right. Y'all are so sweet.
I have a question. Well, first I just want to say this presentation is gorgeous. I mean, it's not often that you see something with such like good, with such clear diagrams, and also like not just clear diagrams, but also like the organization, the color coordination. very, you know, I'm impressed. Um,
Thank you.
I had a couple questions about the essential oils. One is that like you talk about the application of them. What like exactly is the mechanism of application? And then secondly, when you have like when you're testing the essential oils like in tandem, um, what is kind of like, is there like any kind of opportunity cost for using both of them? Like, can a bee only be affected by one essential oil at once, or is like only half of them affected, or is it like what's the like the trade-off for that?
So for the mechanism of applying the oils, I saw in research that there's multiple methods, and we didn't exactly code a specific one. But basically, what happens is they go to the hive and put maybe a pad on the hive and apply the oil onto that pad. The oil evaporates within the hive and like gets onto the bees. And so as time passes, we coded that evaporation would cause the oils to have less of an effect over time.
Oh, and sorry, could you repeat the second part of your question
Oh
again?
yeah,
I
the second
apologize. Yeah.
part was yeah, if you're applying both the spearmint and the thyme, is there like an opportunity cost of that where like the bees can only be affected by one, or they have reduced effectiveness or something like that?
So so at least in terms of our model, we didn't code it in a way that in a way such that like the essential oils would compete against each other. But like I think this in a sense is also like kind of representative of what happens in like real life as well, because at least to our knowledge, like the functions of the two oils are essentially different. One oil affects the bees in a way such that like I think for time it affects um, I believe time
It knocks off
sorry
the mite
Tatiana
from
could
the adult bee, and spearmint only affects the larvae from reducing the transmission rate of the disease.
yeah. And so because like the two oils like essentially have like two different like functions, so I think therefore like it might be safe to assume at least in the lack in this like in the paucity of data, like we can assume we at least for now assume that like there is no conflict between the two oils in terms of function, and so yeah, that's um hopefully that
I
answered your question.
also want to
Oh,
add
sorry.
in that another thing, if we had more time, that we could have looked into is essential oils are pretty dangerous to most animals except for humans. So if we should have, if we had more time, looked into how much oil would like we need, because there's an extent where if you apply too much, then that could lead to the death of bees.
Does it
Yeah,
does it
yeah,
actually
that's
need to
very
be the
thorough. Thank you.
essential oils, or does it, or could you grow these herbs in proximity to the hives? Has anyone looked at that?
So at least for, oh, sorry, do you want to say something first, Tatiana?
You can go ahead.
Oh yeah, sorry. So this is purely based off of conjecture, but based off of like what we talked about earlier with like how the essential oils work. It's essentially like the way that it works is that like the essential oils need to evaporate first for in order for the effects to actually take effect. And so I think the fact that like it's because essential oils are pure like you can kind of assume that they are like pure extracts of the herbs themselves, and the fact that they have to be evaporated on the site means that that like simply growing the herbs nearby in the vicinity of the hives might not produce the same effect as if you were to apply the oils directly. So but that is definitely a good like question to consider, and that is definitely something that if we were to continue this project in the future, we can definitely further pursue down the road.
Okay, very nice job. Thank you.